Doctor, there's something wrong with my dog.
What seems to be wrong with her?
She's having seizures and muscle cramps.
Okay, I'm going to take a look at her. Okay, we're back with the results. We got some good news and some bad news. The bad news is she has Clostridium tetani, which is known as tetanus.
Oh no, what's that?
Don't be worried, it's nothing too serious because you brought her at a good time. So good thing is that she's in the early stages. Now the good news is that this will be treatable and curable. Please, come in my office.
Can you please explain more about the tetanus?
This disease has been dated all the way back until the 5th century BC, but it was not discovered until the 1800s. The disease produces two exotoxins, one called tetanospasmin. It's a neurotoxin that causes the symptoms of tetanus. The most common symptoms are lockjaw, stiffness, problem swallowing, seizures, and muscle cramps, as you have seen in your dog. Cut.
How did my dog even get infected?
Well, tetanus is not something that is contagious. It can be transmitted from animal to animal directly. But it can be found in many places, such as soil, spores in the soil to be exact. Also, it can be found in animal feces or manure. So, in other words, the only way that your dog can get infected or sick is basically by direct entrance of the bacteria. So it has to be something from like a small wound or a cut.
What was the procedure to see what she had?
Okay, so first we take the smear and put it on the plate. We will then prepare the Bunsen burner. We will put a pot over it and we'll put a water gauze over the pot so that it can steam. We will then cover the smear with absorbent paper and set it over the wire gauze. Okay, once the smear is heated, remove the plate off the burner. And then you'll remove the absorbent paper, add some malachite green, we'll rinse off the stain with water. Then we would add some saffron. And finally, we would rinse this off. Then it's ready to look inside of the microscope. We took a sample from her intestinal tract, and then we did something called a spore stain, which is a technique used to stain and visualize endospore bacterium. The results we got showed various similarities to the bacteria Clostridium tetani, which are spore-forming, rod-shaped, drumstick appearance. If you want, we could go take a look at it. This is what it looks like. The rod-shaped drumstick appearance, motile, long flagella, stained red / pinkish, gram-positive.
Can you please explain the ecology of this bacteria?
Yeah, that won't be a problem. Just let me get all my notes and history about it. The ecological role is found in warm, dark areas, usually found in soil or in intestines of animals, just like I told you before. The metabolism is anaerobic which means that it's very sensitive to oxygen, and it's also very sensitive to heat. This is most likely to happen when it's in the vegetative stage, which is one of the life cycles or stages that it goes through. The first stage is vegetative, which then moves on to an endospore or a sporural stage. The vegetative stage is, like I said before, extremely anaerobic and can survive extreme conditions, but it is very sensitive to heat. Unlike the sporural, it is the complete opposite, where it can survive in damp areas such as the animal's intestinal tract and soil as well. So endospores are formed during a process called sporulation. Like I've said before, this bacteria is found in the soil and in the intestinal tracts of animals, even humans, and could be found also in manure and feces. This is very important because it uses these organic compounds as electron acceptors. Clostridia are able to ferment a wide variety of organic compounds. They produce end products such as butyric acid, acetic acid, butanol and acetone, and large amounts of carbon dioxide and H2 during fermentation of sugars. If you'd like, I can also talk about the molecular biology behind this bacteria. They produce different enzymes to externally digest organic material to prepare itself for fermentation. These are special proteins that are needed for its survival. The bacteria contains 2,372 ORFs, which are specific sequences for specific proteins. Only 61 ORFs are encoded for the tetanus toxin and collagenase. They have a virulence factor of creating toxins which hurt the host, and another virulence factor involves destroying the tissue of the host. PE88 harbors the genes for the tetanus toxin, and CTP33 encodes for collagenases, which is the protein that involves destroying tissue of the host. Obviously, the bacteria is a pathogen, and that is something that infects the host and it could later lead on to death. The mode of action of the tetanus toxin produced by the bacterium is now well understood compared to the 1800s. The tetanus toxin blocks the release of the neurotransmitters from presynaptic membranes of inhibitory neurons of the spinal cord and the brainstem of mammals. It catalyzes the proteolactic cleavage of the synaptic vesicles protein synaptobrevin. This leads to continuous muscle contractions, which are primarily observed in jaw and neck muscles, which is why you are seeing lots of muscle cramps and seizures. It is important to know about this organism because it allows you to recognize the symptoms, and it also allows you to know why it is important to get the vaccine. So I can't not wait until your dog is healthy and wait to see both of y'all be happy.